Today, let's understand specific immunity, or it is also known as adaptive. Let me write it down. Adaptive immunity. We have already covered the innate immunity. In this one, we will be covering specific immunity, or also known as adaptive immunity. First feature of this is it is acquired. Means you are not going to get it naturally. And our immune cells, like you have B cells, you have cytotoxic T cells, they're going to actually gain this particular immune response by, getting exposed to the pathogen, right? And it's also anamnestic, means it's going to have that memory response. So if there is re-exposure of the pathogen, it's going to initiate the response very, very quickly. So if you have a pathogen and first time infection, immunity will be generated, and then pathogen is basically eliminated. So let me write it down. Pathogen is gone. Pathogen is gone, right? But second time, if the same pathogen tries to enter, then immediately the adaptive immunity, which is including the B cells, antibodies, T cells, they're going to immediately attack on this particular pathogen and try to stop it. The third point is they're going to complement the innate system. So adaptive immunity actually strengthens our innate immunity. So innate immunity and you have direct link with the adaptive immunity, and adaptive immunity will try to basically strengthen, and innate immunity also try to strengthen the adaptive immunity, right? So they basically complement each other. Fourth one is cooperation. They work together. Adaptive immunity and the innate immunity, they're gonna work together. So let me write it down. Specific and adaptive immunity, right? So the specific one, which is adaptive immunity, and plus you have non-specific immunity, which is innate immunity. Both of them, they're gonna work together so that our immune system will work against the pathogen. Got it? Okay, so next point is, what are the important functions of adaptive immunity? So let me write it down. Major functions of adaptive immunity. Correct? So that will be the topic that we'll be discussing. The first one, very, very important function, is the recognition. How the immune system is going to recognize. So basically, if you have a pathogen versus the host cell, innate immunity is not going to understand that, but adaptive immunity will differentiate between our self component versus non-self components. And based on that, it's going to respond. So that's very, very important feature of the adaptive immunity. Second one is the response. So you have target pathogens, right? And you will have immediately those T lymphocytes, B lymphocytes, they're going to proliferate, and immediately these particular cells, they're going to amplify, right? So let me write it down. Amplify specific lymphocytes. So all those lymphocytes, they're going to immediately amplify and then work against the pathogenic molecules. And they're going to target that particular pathogen, which is present inside the body, targeting the pathogen. So that is the response. The third one, very, very important one, is memory, which is anamnestic. I've already told you, right? If there is re-exposure of the pathogen, if pathogen is, pathogens, they are very clever. They try to re-enter into the body, then more intense, and as well as the faster as compared to the first exposure, response will be there, as well as it's going to prevent the disease. And that is the idea of vaccination, right? So we initially try to expose our, I need to change this slide. Yeah, I'll come back. So pathogen is basically, and vaccine is basically to mimic what the pathogen is trying to do in our body, right? And then we give vaccines. Okay, next is for major characteristics. What are the major characteristics of this immune system? One is self versus non-self. It is going to quickly differentiate between what are the cells that are of our body, which is self, and what are the non-self components of the body, and they will be destroyed by these lymphocytes, and they will work very, very effectively against that. Second one is diversity. Can you imagine so many pathogens are there, and we have the system that will create antibodies, these Y-shaped molecules, antibodies against all those pathogens. So you have basically, second one is T-cell receptor also, because that will also bind to antigen, right? So trillions of antigens can be recognized by B cells as well as our T-cell receptors. So B cell receptor or antibodies as well as T cell receptors, right? Another one is specificity. Adaptive immune response is highly specific. Innate is not specific. So pathogen, it is going to get targeted against the pathogen. Let's say this is pink color pathogen is one pathogen, right? You will get the adaptive immunity. Then you have another pathogen, which is, let's say, green in color. So this is pathogen 2, new pathogen, and it will get the adaptive immunity, let's say 2. The second adaptive immunity against that one, right? So there will be like, it's not going to cross react, right? So 2 adaptive immunity is not going to work against the first one, right? So that's very important point. Memory, as I've already told you, that if pathogen is trying to re-enter into the system, then it's going to get eliminated very, very effectively because the response will be very, very quick, right? So if try to re-enter, it's going to get attacked by the adaptive immune system. So that's very, very important point. Okay, let's move to the next one, which is arms of our adaptive immunes or specific immunity, right? Arms of the specific immunity, very, very important. Next is, first one is humoral. Humoral is linked with the antibody associated response. Humoral, antibody mediated. Second, cellular, cell mediated, right? And humoral immunity, let's understand humoral immunity in detail. You have in humoral immunity, it basically is antibody associated. So you have B cells, right? B lymphocytes. These B lymphocytes, they're going to produce these magical molecules, these Y-shaped magical molecules called antibodies. Antibodies are soluble glycoproteins. They are soluble glycoproteins. They're present in body fluids, right? And what they basically do is, because they are soluble, they can immediately bind to these antigenic molecules, these pathogens, and the antigen present on the bacteria, on the virus. They're going to try to actually opsonize and neutralize the virus and bacterial particle, so they become susceptible to phagocytosis for immune cell attack, right? So microbes, including toxins, including viruses, all of them are susceptible for the attack by the antibodies. Antibodies are wonderful molecules. So they will kill directly. One point is that they will also tag these pathogenic molecules and perform the opsonization using immune cells, and also complement based pathways also. Complement will also act on those opsonized molecules. So it's very detailed process. Next is cell-mediated immunity. Let's talk about cell-mediated immunity, which is T-cell mediated. So let's imagine this is our T-cell, and T-cell is looking for another cell, which is host cell, but it is virus infected. So can you imagine our T-cells, they have the ability to recognize these virus infected, pathogen infected cells, and this cell, then T cell specifically, is going to become cytotoxic T cell, and that is going to basically bind to this virus infected cell with different receptors. We'll have T cell receptor working over here, we'll have co-receptors working over here, and it is going to target this host cell because it got the signal that this is infected. So we have to take a call and kill our own cell. So cytokines are going to get released. Innate immunity, that is the non-specific one, is going to go up. Adaptive is going to go up, and then it will try to kill that host cell and also try to protect our other cells, right? So T cell basically, can you imagine, T cell has a wonderful capability to recognize self versus non-self and self that is infected with viruses. So it can recognize all of them and then kill the problematic ones. So T cell should have, and must have, I would say, must have ability to recognize self versus non-self versus infected self cells. Very important property, right? Okay, now let's move on to the another part, which is very important part, antigens. What are antigens? Antigens are nothing but they are proteinaceous molecules, right? They can be proteinaceous, they can be carbohydrate, they can be lipid, but they can cause immune response to get generated in the body. They can react with antibodies, and they can also react with T cell receptor. B cell receptor is nothing but the antibody, okay? They have the ability of immunogenicity. Immunogenicity is basically they can induce immune response. So proteins are there. You have nucleoproteins, right? And then you have polysaccharides, and in the last you have glycoproteins. So this is the ability of immunogenicity of all those molecules. Proteins usually have the highest immunogenicity, and the glycolipids usually have the lowest immunogenicity. Now, if we talk about size, it's approximately 10,000 or more than that Dalton molecule, larger molecules, right? So smaller molecules, they are not going to generate the immune response, right? That's why they are also known as haptens. They are not going to generate the immune response. And if you talk about another one, which is foreignness. So antigen should have the ability to be non-self molecules, right? Some small molecules cannot actually initiate the response. Highly conserved molecules also, they are also not recognized as foreign in many cases, right? So if the molecule is highly conserved, then it's not a good candidate for vaccine also. All right, now let's move on to the next one, which is again very important topic, which is epitopes. What are epitopes? Epitopes are antigenic determinant sites. In antigen, you will have different molecules. Antigen is a very big molecule, right? But inside that antigen, you can have multiple different types of sites or sequence of amino acids, specific regions that are basically binding to the antibodies directly or with T cell receptor. Those are the epitopes. They are usually present on the surface region or in the terminal residues also. Very important point, right? If we talk about chemistry, so these can be sugars, that is glycans. These can be amino acids, very important point. Side chains of molecules, aromatic groups even, okay? And then organic acids. They all connect as epitopes. And then this is the one which actually binding with the antibody. Valence. Valence is how many epitopes copies are present in the antigen. That is the valency of that particular antigen, right? For example, as you can see, more epitopes are present, so that means much stronger immune response can be generated. So this is how you can have a vaccine candidate, you have multiple epitopes, you will get better response. Okay, now let's talk about affinity versus avidity. Okay, what is the difference between affinity in case of antibody versus avidity? So affinity, antigen antibody interaction, that is what we are trying to understand. Affinity is binding strength at one antigen binding site, a specific antigen binding site. What is the strength of that particular antigen antibody binding interaction? Increased during a response. So basically, whenever there is response, it is going to get increased. It's also known as affinity maturation, right? In case of monovalent antigen, you will have single binding site, right? And this is technically, this is measured by, we'll not go into the detail, but let me tell you that it is measured by equilibrium dissociation constant, KD. So it will determine how strongly it can bind to that particular site, right? With the diagram, if I try to show you, let's imagine this is the surface where you have epitope or antigen, which is monovalent antigen, right? And then antibody is binding over there. And let's suppose this is the monovalent antigen. What is the strength of this interaction? And that will determine how good is your epitope. Next is avidity. Let's understand avidity very, very quickly. All sites combined, all the antigenic determinant sites, they combined on that antigen will contribute for avidity. So total strength of all the sites that are present on the antigen where all the different antibodies are binding. So that is true or applicable basically for multivalent antigens, right? And multivalent cross-linking will also be determined by measuring the avidity, right? So, and this also will lead to the complex or precipitation and how quickly it can precipitate, because you'll have multiple interactions going on, and this will lead to the precipitation or complex formation. Now, determine the functional antibody activity in vivo. So, this is also very, very important to determine the functional activity. Now, let's say this is the antigen and you have multiple antibodies, and this will lead to the complex formation because you have so many antibodies and so many antigenic determinants working together. Okay, now let's talk about small molecules called haptens. And these are the small molecules. Let me write in small molecules. They are non-immunogenic. So most of the phytocompounds and all, our immune system is not going to work against those. And if you put them and tag them in carrier proteins, so you have carrier, let's say one carrier protein, and you tag haptens on top of that, they become immunogenic. Interesting process, right? And in that case, you have epitopes and immune system is going to detect it. It is going to become epitope. Okay, but if it is independent, immune system is not going to detect it. So let's imagine this is the CP, carrier protein, and you have these haptens attached on those heads. So B cell is going to produce and T cell also. B cell is going to produce the antibody, and T cell is going to produce the T cell response against it. Wonderful example is of penicillin. Okay, penicillin is a small molecule. Penicillin is not going to produce any kind of immune response, but if it binds to the serum proteins, some of the serum proteins, they have the ability, and you can see this white colored bead is bound to that red color serum protein. It can strongly initiate type 1, type 4 and type 4 hypersensitivity reaction, and it becomes deadly. It becomes dangerous because of that. It's also known as penicillin allergy. So with hapten, I hope you were able to understand how the penicillin allergy has been in our body has been initiated. CD molecule. Let's talk about cluster of differentiation molecules. So in cells, we have these CD molecules present on the surface. Okay, on the surface of immune cells, they are usually membrane proteins, and we also call them receptors. So let me write it down. Receptors. Right? Membrane proteins. They are also known as receptors. They can induce very, very strong immune response. Right? These are really important for actually interaction of the cells with other cells and induce the immune response when exposed to different lymphocytes. So a lot of lymphocytes, they require these CD molecules. These CD molecules are also important for cell identity, that what kind of cell lineage we have, whether it is T cell, B cell, what stage of maturation the cells are in, whether they are activated or not. This is being determined by these CD receptors. Very, very important. They can be marker of those stages. So CD4 is the marker for HIV receptor, or it is going to bind to the HIV receptor. Next, CD38, stem cell marker. Okay, and you have approximately 300 characterized CD receptor in human body. So huge diversity is present in our case, and so many CD receptors are there, many are still unknown. So let's talk about some of the selected CD molecules and their functions. Okay, CD3. Let's start with CD3. This is very, very important for T cell signaling complex. So for T cell signaling, it's very, very important, right? And T cell activation signal specifically when it goes to internal side, CD3 is required. CD4, it's the receptor that binds with MSC class 2 molecule. Please, everyone remember that. Okay, next is co-receptor on T cells. This is the receptor which is present on T cell and specifically in helper T cell, right? HIV primary entry receptor is CD4. I told you earlier, right? So CD8, MSC class 1. CD8 is the MSC class 1 receptor. It's going to bind with MSC class 1. So co-receptor for cytotoxic T cells. Okay, it's a co-receptor for cytotoxic T cell. CD4 cell count becomes a marker for HIV infection. It is a marker for human immunodeficiency virus. It's also disease is also known as AIDS. So if you have CD receptor which more than 200 cells, right, per microliter, that is the indication that AIDS could be there. CD34, you have the stem cell marker. Then you have CD54, which is an NK cell marker, right? You have CD19/20, which is B cell marker. There are so many. I want you to please go and remember most of them. Some of them I've already included. But I'll try my best to actually include those things in near future, so that we can devote like a small lecture to every CD receptor, so that we can understand what are their functions, right? All right, so I think that was the introduction for specific immunity, which is antibody mediated, and also our cell mediated immune system. And now we have started the cell mediated. We are finished with the innate immunity course. So please go and watch the entire innate immunity course and come to the adaptive immune system course and PPTs, as well as MCQs and notes. They are available, as you can go to the description and just click and download and play around, prepare your own modified notes practice the MCQ quiz, download the PPT and see whether you can basically use this for your own teaching, right? And it becomes a resource, open resource for everyone. So if you like the contribution that I make for sciences, for teaching, please do subscribe to the channel and also share the video with your friends, whoever is studying in this particular domain. And if you have any doubt, please use the comment section. I will try to answer those questions to the best of my capabilities. All right. So we'll meet in the next one. Till then, take care, everyone.